They'll be coming in, but you go ahead and start. Don't wait for them.
All right, well, hi, thank you for coming to our poster session. My name is Omar Khan, and I have Jennifer Nguyen and Leonardo Tenorio with me. And we've been working on a project called Expressing the Recombinant C-Terminal Fragment of Pili from Myxococcus xanthus with Dr. Bowden at the University of Houston downtown. So we expressed the recombinant C-terminal fragment of Pili in Myxococcus xanthus. Specifically, we wanted to study the interaction between the type 4 Pili and the exopolysaccharides that the bacterium uses to glide across surfaces using S motility. Leading up to the semester, we did the following. In order to study this, we needed to clone the C-terminal fragment of Pili, and this involved designing primers in silico based on our target C-terminal fragment and the plasmid PPE vector down here. Determining the restriction enzymes that would be best, and finally running a PCR to obtain copies of our target DNA fragment. We ligated these fragments into plasmid vectors, which we ended up using in this semester. Leonardo will now describe what we did with these clones.
Okay, thank you. After selecting clones 4, 6, 7, and 8, we proceed to perform a mini induction. The clones were grown for about 2 hours to mid-log phase. Then the clones were induced with 225 micromole of isopropyl beta-thiogalactopyranoside, or IPTG, that mimics allolactose and binds to the LAC repressor to free the promoter, and transcription of the target protein can start. Furthermore, the cultures were incubated for 3 hours at 37 degrees Celsius. We pelleted those cultures and resuspended them in saline buffer to concentrate the expressed protein while removing contaminants. Additionally, the extracts were analyzed with SDH page and Western blot. As you may observe in the figure on the top left, that will be the SDH page gel figure. The desired recombinant proteins was observed in induced clones 4 and 6 with a band size around 18 kilodaltons. Jennifer will discuss the Western blot. Thank you.
Thank you, Leonardo. So the first step of the Western blot, as Leonardo described, was the gel electrophoresis, and this was to separate the sample's proteins. The next step after that is we're going to transfer those separated proteins out of the gel into a nitrocellulose membrane. Now, on the N-terminus of the recombinant protein, we introduce the anti-histidine antibodies that bind to the 6-histidine. Our recombinant protein was detected with the anti-hist tag antibody in the bottom left image. You can see that in lanes 4 and 6 induced. This confirmed the expression of the C-terminus of PIL-A protein. Thus forward, we will be calling that PIL-A-CT. So overall, in the column labeled expression of recombinant PIL-A C-terminus, we confirmed the presence of our recombinant protein using the SDS page gel and Western blot. So our next step was to do a large induction of PIL-A-CT4 and PIL-A-CT6. We started by inoculating a large quantity of LB agar and induced protein production with, as Leonardo stated earlier, IPTG. This induces beta-galactosidase activity in bacteria to detect LAC gene activity in cloning. We ran the cultures in a nickel-charged column using avidity chromatography. The nickel binded to the 6-histidine tag on our proteins, and the bound protein was eluted with increasing concentrations of imidazole. Each fraction allowed us to measure how much protein was being eluted in each concentration. Ideally, the last dilution should have had all of our isolated proteins. However, the recombinant protein can be detected in the 10% elution, which is seen in the top right image. We hypothesized that our protein is dimerizing because bands that we see in the 10% elution are much larger than our target. The 10% elution bands are seen around 30 to 60 kDa, and our target band was at 18 kDa. After performing the semi-dry transfer, the gel was placed in Kamasi blue dye And this dye was used to dye the remaining proteins to confirm that our target protein has transferred, and we didn't want to transfer over the larger proteins. So on the bottom right image, the bands that you currently see are the larger, heavier proteins, and our target protein has transferred. Now my research partner Leonardo will be explaining what we did next.
So then we moved to the electroporation into the E. coli GM101 strain. So considering the results, we prepare electrocompetent cells of E. coli GM101 by harvesting them mid-log phase cultures through centrifugation and washing them 3 times, which double distilled water at 4 degrees Celsius. We then resuspended the culture in 10% glycerol. Furthermore, the cells were electroporated using the plasmids extracted from cultures with the clones 4, 6, 7, and 8. The electroporans were plated in LB medium that contained ampicillin. And lastly, the plasmids were extracted and digested using HIMD3 and BMH1 restriction enzymes, as you may see in the bottom left figure. The digested plasmid will be loaded in agarose electrophoresis gels to confirm the presence of such plasmids. Omar now will discuss the future directions.
So the future directions are we're going to do a mini induction using our new E. coli strain JM101, which will hopefully express our recombinant protein in greater quantities. And we will use affinity chromatography to, again, try to purify these proteins and run a binding assay to see how it binds with extrapolysaccharides. And I'd like to thank the Scholars Academy and the DOED MSCIP for funding this project, and Dr. Bowden for leading the project.
Thank you.
Thank you.
Great. Can we ask a question?
Sure.
Okay, so what does this get us closer to? Can you answer in terms of helping humanity and or less than humanity? Tell me how it applies. What will we've learned we can do this, but then what?
Who wants to answer that one?
I think that just the techniques that we're using and being able to figure out how these proteins are able to bind and how the motility occurs in the bacteria would be beneficial.
Just previous studies find out this relationship between the peptide and how it glides, because in ecology, how it glides using the peptide and everything, but that was qualitatively, and we're trying to quantify it to make sure that it does, and it's involved.
Thank you.
You're welcome. Thank you.
Great, guys. Good job.
Dr. Bowden.
Thank you. Hi.
Thank you.
Okay, I'm going to leave you. You can start again. Maybe they didn't hear the first part, or, if they want to choose another. Stay in your room and keep